Okay, welcome back again. Our next speaker is Cécile Carrère from Paris again,
yeah.
And she's talking about modeling tumoral heterogeneity for chemotherapy optimization, optimal control, theoretical and numerical analysis.
Thank you for the introduction, and thanks to the organizers for letting me speak today. And I'm really enjoying Rio, as you can see. So today what I will talk about is some work I have been doing during my PhD thesis. Now I'm a postdoc in Paris, so I have to keep all the different logos. So I'm going to talk about tumoral heterogeneity, and especially how to cope with resistance to chemotherapy. So when you start treating a tumor, it can happen that the tumor begins to become resistant to the chemotherapy. So in many models, and in many biological models especially, this resistance is taken into account as a progressive phenomenon. So you start with cells that are completely sensitive to the chemotherapy that you're using. Then when you begin to treat them, to use the drug, you kill a lot of them. The one that survived the attack began to accumulate mutations. When you continue treating the tumor, it accumulates more and more mutations until the cells that you end up with are completely resistant to the chemotherapy. But this accumulative mutation is quite a long process compared with the timescales that we deal with some types of resistance to chemotherapy. So to explain this other phenomenon of resistance, a new idea is to consider that the heterogeneity is in fact already present in the original tumor. That before you begin to treat the tumor, there are already cells that will respond to the treatment, that will be killed by the treatment, but other cells that will not see it, that will be already resistant to it. So when you use the same schedule of treatment on this type of cancer, then using high doses of treatment kill all the cells that were sensitive to the drug, leaving only the resistant cells who no longer have competition with the original cells. Thus, they can begin dividing up and invading the original space that is left, and we end up again with a big resistant tumor. So now the question is this: with this new hypothesis, how can we still treat the tumor? How can we reduce the tumoral charge without the tumor becoming resistant to our chemotherapy? So to do this, I will first show you some in vitro experiments that we based our model on. Then I will show a brief trajectory studies of this problem, and I will treat the problem with two different theoretical theories, with two different mathematical theories. First, the optimal control, and then dynamic programming and Hamilton-Jacobi equations. So first, this series of experiments that we studied were realized in the CRO2. It is a pharmacology lab in Marseille. So it was realized by Manon Carré and her team. They study in vitro tumors. So inside a petri dish, they mix two populations of cells, one that is sensitive to a certain drug and the other one that is resistant to it. In order to follow the two different populations, they were able to mark fluorescently the two populations. So the red ones are sensitive to the drug, and the green one are resistant to it. So what they do is they begin to the co-culture with the two types of cells, and they let them evolve in the Petri dish under different schemes of treatment. They can follow the spatial distribution of the cells during the experiment. They can also do close-ups to see if the cells are clustering during the treatment, under the treatment. And they can use fluorescent measuring to have the total number of both populations. So what happens under the different treatment that they were using? If you give no treatment to your Petri cells, to your Petri dish, you see the sensitive population that begins growing and growing, filling up the Petri dish, and then it's blocked and it arrived at a confluence number, so it completely fills the Petri dish. And for the resistant population, they do survive. There are still some resistant cells, but there are really few of them. It's difficult to see, but here there is a gap in the scale, so there are really, really a lot less resistant cells than sensitive cells. Now, they used a scheme of treatment where you give huge amounts of drug at really under a really short period, and you wait a long time before giving another dose. So this is to imitate the MTD treatment that is classical in chemotherapy, where you give huge doses of the drug in order to maybe swipe all the cancerous population the fastest possible. So if you do that, you do kill all the sensitive cells really quickly, but then the resistant cells don't have any more competition, so they start dividing up and filling the Petri dish. And then they tested another scheme of treatment where you give smaller doses of treatment, but more regularly. And what happens is that you kill a lot of sensitive cells, but not all of them. And the number that remains is still enough to repress the resistant cells. So in this situation, we have something that is quite interesting medically. It's a tumor that is relatively small and globally sensitive to our drug. So this is a really interesting situation. So now that we have these different experiments, we propose a model for that. Because it's a simple biological experiment, we propose a simple mathematical model with the two types of cells, S and R, that evolve during time. They obey a logistic growth equation with here the competition that regards both of the populations, both of the type of cells. Here, M is a size ratio, meaning that the resistant cells are in fact a little bigger than the sensitive cells. We saw that resistant cells really have a disadvantage in competition with the sensitive cells. So we add this competition term in our model. And finally, the drug that is represented by C here, the concentration of the drug, only acts on the sensitive cells. So with that, we want to first test that we do represent the different experiments, and then propose protocols that will attain the objective in some sense. So first, to convince you that our model do represent all the situation, if we look at our model in the phase plane, so here is the population S, the sensitive population and here the resistant population. If we do not give any treatment, in this phase plane, the only attractive stable point is this one, with only sensitive cells at the maximum capacity of the Petri dish. And it does correspond to the experiment where we gave no treatment at all. Then, if we give a weak dose of treatment, a low dose of treatment, we have the operation of two attractive stable points. The first one is the interesting one with fewer sensitive cells and no resistant cells. But there is another, and this corresponds to the situation here with low dose of chemotherapy. And the other point is here with only resistant cells at the maximum capacity of the Petri dish. And we still have this if we give a strong dose of treatment. So here the only stable, attractive stable point remaining is this one with only resistant cells. So this was just to convince you that qualitatively this model is at least okay. But now that we have this, we want to use mathematics to propose new protocol for chemotherapy. So first, an idea that is quite frequent in this kind of study is using optimal control. To do that, we propose a cost for one experiment. So we propose this following optimization problem. Given an initial population and a finite time of experiment, we want to minimize this cost. This cost is composed of two parts. First, the size of the tumor at the end of the experiment, and it wouldn't be relevant to just use that as a minimization, because otherwise you just do nothing and then just before the end of the experiment use a large dose of treatment. Not interesting. So we add this integral cost here, where we see the integral of the size of the tumor during the whole experiment, and we put the two populations to the square so that we really penalize the tumor that grow at a high population. And the coefficients A and B are just tuning coefficients, but we can change them whether the medical teams will want us to emphasize on the resistant population or not. So now that we have that, we can use the optimal control theory using the Pontryagin minimum principle, which tells us that treatment that is optimal for this cost will also minimize the following Hamiltonian, where P1 and P2 are an adjoint vector here, which we have the equation of. This looks quite overwhelming, but in fact reduces to a really simpler expression, because C here only appears in this term. So if you want to minimize H, then it all depends on the sign of P1, because alpha and S are positive. So if P1 is positive and you want to minimize this, then you just have to set C to its maximal value. If P1 is negative, then to minimize this, you set C to its minimal value, so 0, no treatment. And if P is not on an interval, you can go into the equations and toggle with them and end up with an expression for the treatment on this interval. In the optimal control theory, in this framework, this corresponds to a singular arc. And we can do the parallel with the intermediate doses of treatment that we were seeing in the experiments, and that are called in medicine metronomic treatments. So low dose of chemotherapy during a longer period of time. So these are just necessary conditions on the treatment, but we cannot say for the moment that we do have the operation of singular arcs, so of metronomic treatment, in our optimal chemotherapy. So to investigate that, we did some optimization in silico, so numerical experiments, and we decided to minimize the same cost over a cycling treatment using either no treatment or the metronomic treatment, in fact, the singular arc we defined earlier, and some maximum tolerated dose, so the maximum dose that we had in the definition of the treatment. So, and what we see is that here in black is the treatment over time, and in red the total number of cells. We see that we do have some singular arc appearing, and that it really keeps the population at some kind of stable level. So what you do is, at first, when the tumor is really small, you don't start the treatment, letting thus the sensitive cells gain enough power to begin really oppressing the resistant cells. Then, when there are enough of them, you can start the treatment at the singular value so that the cells are stabilized around this level. And then, after some time, you can kill almost all of them by using a small burst of maximal dose. Then you end up with a really small tumor that maybe is mostly resistant, so you have to, again, let the tumor evolve without any treatment for a while before beginning a new cycle. We can compare it to the same kind of cycle, but without singular treatment. If you don't allow the treatment to go to the singular value, so if you just do either no treatment or maximal treatment, in order to minimize the same cost, you are forced to let the tumor evolve at much higher populations. So here, we were stuck under this number of cells, and here they can attain 3 times the size. So, well, there are also some advantage for this treatment, because here we are really often under a certain number of cells, but imagine also it is really regularly growing and collapsing, growing and collapsing. So there are some advantages and drawbacks, and maybe this one treatment is more interesting. And furthermore, if we look at the resistance, the proportion of resistant cells inside the tumor at that time, we can see that here we're really reducing the proportion of resistant cells during the whole experiment, while here, when we don't allow singular treatment, we are really often at 10% resistance inside the tumor. So this first work was published in Journal of Theoretical Biology this year. Yeah, that was cool. And now I'm going to talk about another problem. If we look here, I said several times that we are stuck under a certain threshold, but this was not something that we asked for the optimization. This is completely a not really asked phenomenon inside our treatment. So, but it is in fact what is more interesting for the medical team to decide a certain threshold that over this size threshold, the tumor will be dangerous, and under this size threshold, well, we can manage this tumor. So to do this new problem, we can look at another theoretical framework which will be the Hamilton-Jacobi-Bellman equations. So we define two new problems. The first one is a viability problem. So we define with the medical team a certain size threshold, and an initial tumor will be defined as viable if there exists a treatment such that the size of the tumor can be kept forever under this size threshold. So our problem will be to determine the viability set NQ, which means the set of all initial tumors that we can assure that we will always keep under this size threshold. But nonetheless, it can happen that at the beginning of the treatment, we encounter a tumor that is maybe too big or maybe too resistant to be viable, but that we can still treat by an initial treatment. So the secondary problem will be the reachability problem. If we have an initial tumor that is not inside the set NQ, bring it inside the set NQ as fast as possible, but in no longer time than big T. So we set a maximal time of initial treatment, and we want to see which tumors can be brought inside the viability set in this time, and if possible, to minimize this time of entry, this time of initial treatment. So if we go back to the phase plane of that was previously, this viability problem means that if we set this size threshold, we want to see which tumors that begin here can be kept here inside this triangle. So as I said, it can be possible that even if we start inside the triangle, all the trajectories from here all go out of the triangle at some time. So for example, this initial tumor is not viable in our definition. So the viability set will look something like this. And then we want to see which tumors are inside this reachability set, which one can be brought inside this in a reasonable time. And we want to minimize the time of entry, the time of the initial treatment. And all these problems have a nice translation inside the Hamilton-Jacobi-Bellman framework. Because if we define, we will define first this value function Q. So what is it composed of? For a certain initial tumor, it is the minimum over all trajectories of the maximum of this quantity. G is a function that is negative inside the small triangle and positive everywhere else. So if you take a trajectory that goes outside at some point, then the maximum of this quantity will be strictly positive. And if all the trajectories starting from this point go outside the triangle, then V will be positive at this point. Thus, the sign of V completely characterize our viability set. And if we toggle a little this equation, this definition, we can see that V is in fact a viscosity solution of a certain Hamilton-Jacobi-Bellman equation with obstacle. This way, we can solve it numerically and thus determine numerically the viable set. And there is something similar for the reachability problem. So we define another value function that will characterize both the reachability set and will be able to give us the optimal trajectories. And this value function is also a solution of a Hamilton-Jacobi-Bellman equation. And numerically, what we observe, so here in blue we have the viability set for a certain value of the parameters. We have some trajectory that are shown here. So for most of them, what we observe is that we have to let them evolve for a while without treatment. Here is represented the treatment corresponding to those trajectories. So the green one, for example, has enough sensitive cells, so we can start the treatment at the very beginning at its maximum value and bring it really quickly here inside the viability set. But the others don't have enough sensitive cells. They are too high in this diagram. So you have first to let the system evolve without treatment. For the black one is for quite a long time, until it has enough sensitive cells to begin oppressing the resistant cells. And then we can start the treatment and bring them inside the viability set. This part is still a work in progress with Hasna Zidani, and it will involve other problematic, and especially how to reconstruct the trajectories once we are in the viability set. So once we are sure that we can control the tumor and keep it under a certain threshold, how do we define this optimal, this optimal treatment? And this is the treatment that will involve, numerically we see that it involves mostly intermediate values of the treatment. So just the treatment. So this, all that I've shown really confirmed the importance of metronomic treatments to cope with tumors that are heterogeneous. The medical team we worked with, the experimenters we worked with, accepted to test the optimal control solution that we shown. It didn't work, but it's probably mostly a problem of modeling. Probably the effect of the treatment is not well taken into account in our model. But nonetheless, this gave a nice framework for future work. So if we adapt the model to the new experiments, we will be able to do the same theoretical approach. Meanwhile, on the biological side, they studied why there is a repression of sensitive cells on resistant cells. It has to do with their metabolism. They don't create lactates at the same rate, but I can give you details afterwards, but it's not my work. And they want to begin experiments on the same tumors, but now encapsulated inside same tissues, still in vitro, and then pass these experiments in vivo, in mice, to see if we can still have this advantage of using metronomic treatment on those heterogeneous tumors. And for me, on the biological side, so we could adapt the model to the new experiments and take into account the same tissue. The immune system also will be something else. What I've shown you is also really idealistic, because I suppose I have complete information on the number of cells, both sensitive and resistant. So it will be important to adapt this modeling to partial information if we can only have the size of the tumor, for example, and at some point in time. And during my postdoc, I'm currently studying the mechanisms of resistance appearance, and especially if we give several drugs, will the tumor develop one single resistant clone or different clone resistant to each drug? So, thank you for your attention.